So welcome back for the second part of the cells lecture here. Um, as I said kind of at the end of the very last lecture, I went ahead and split this lecture up into two different lectures. And the reason being is it was just so long, so I wanted to make the files just a little bit shorter for you guys, and hopefully then they didn't take quite as long for you to download. So again, this second part of the cells lecture, again, this is still chapter 3, is going to focus more on what we call membrane transport, and that's going to be how we get things into and out of the actual cell. And then the very last part of the lecture, we'll actually talk a little bit about uh, how cells will go through division, and then how cells will process energy. So that'll pretty much, again, carry us through the rest of chapter 3. So again, we're going to start off here by talking about how we actually bring things into and out of the cell. And this is what we're talking about when we talk about membrane transport. So membrane transport is again, a way that we bring things into and out of the cell, and we're going to talk about the fact that membrane transport can actually be what we call unassisted, meaning there are some things that will just simply move or diffuse across the membrane. So we'll talk about some examples of this being things like oxygen and carbon dioxide. And so as you can see here, diffusion again is what we call kind of a passive process. And what that means is that it doesn't require, oops, I'm sorry, I just crossed out passive for you, but what it means is that it doesn't require any extra energy, okay? And so literally things are just going to be moving down what's called their concentration gradient. And so the formal definition for this would be, again, the random movement of molecules down their concentration gradient. And in this case, you all probably intuitively are very familiar with diffusion. And I always give this example when I'm, actually inside a classroom. I tell the students that if for some reason, let's say, I had a, skunk up on my podium, and that skunk were to spray me and release all these lovely odoriferous particles into the air, everyone intuitively knows about diffusion, because most people in the room are not just going to hang out in the room to see what happens, right? You know that intuitively those odoriferous particles that might start in a very high concentration around my body are going to move down their concentration gradients, right, into the back of the room where they're in a low concentration. So again, many of you have experienced this. Or I also give the example of, you have, if you have dogs, I have dogs, as you probably could tell in a few of my lectures where they tend to bark. If you have a dog in the room and they're, sitting near you, and all of a sudden you hear them fart, you know that those lovely odoriferous particles are going to move from their high concentration, where they've just been released from your dog, down the concentration gradient to in a lower concentration surrounding your nose. So again, we all intuitively kind of know what diffusion is. And so that's exactly what it is, just the movement of molecules down their concentration gradient from an area where they're in a high concentration to an area where they're in a low concentration, until those, again, molecules are in what we call equilibrium. So, oops, I'm doing a horrible job of drawing the slides here today. So again, that's basically what diffusion is. And again, diffusion can occur, okay, right across our permeable membrane. So our cell membrane, okay, is what we call a semi-permeable membrane. And what that means is that it's going to allow some things to pass through the membrane and some things will not. So, okay, there are certain substances that can diffuse by passing right through that lipid bilayer. Okay, now I'm just going to go ahead and kind of introduce this now. So, things that can pass through that lipid bilayer by diffusion, meaning things that will just simply diffuse directly across the membrane, are going to be things, okay, that are typically small and uncharged. And the reason that uncharged molecules, okay, things like lipids, which carry no charge on them, are going to pass through is because remember there's that the middle of your sandwich, the lipid portion of your lipid bilayer, okay? And because you have those fatty acid chains in the middle of that membrane, then those fatty acids they are lipid based and they don't like to interact with other things that are charged. So remember, they don't like to interact with water. They're what we call hydrophobic, and they don't like to interact with anything else that's charged. So things that are small, okay, and things that don't carry a charge, okay, and that are not charged, are more likely, again, to diffuse across the membrane. Now, I think it's easier to remember, okay, what's not going to cross the membrane, okay? And so the things that will not easily cross your membrane, your cell membrane, are going to be, and I'll remind you of this a little bit later, are going to be things that are large and charged, okay? So that's my way to remember. Things that are really large molecules, and really charged molecules are not going to want to pass through that lipid bilayer. So again, let's talk about some examples of diffusion in the human body. Again, when we talk about simple diffusion, I kind of mentioned this. Oxygen, okay, and carbon dioxide are going to be two gases that will simply diffuse across membranes. So again, they're going to move right across the membrane, right down their concentration gradient. And this is a good thing because this allows us to exchange gases quite rapidly, right? So this allows us to exchange these gases between the blood and the air in the lungs quite rapidly. So oxygen and carbon dioxide are going to be two molecules. And again, these are small molecules, okay? And they, again, are not going to carry a charge on them. So these small molecules that don't carry a charge on them, or again, much of a charge, are going to pass right across the membrane. And then we're going to talk about certain substances that move across the kidney tubules. We're going to have certain substances that will directly diffuse right across the kidney tubules, and you'll see we'll talk about that when we get to the kidneys. And then the diffusion of sodium and potassium ions can actually occur through open channels in the plasma membrane. Now we're going to talk about, if you look at this word here, they say the diffusion of sodium and potassium ions, okay? And that tells you that if it's an ion, that means it's charged, right? It's either gained an electron or lost an electron. Remember that from the chemistry chapter. So in that case, again, simple diffusion right across the membrane would be much harder because things that are charged don't like to move across that membrane. However, sometimes, and we're going to talk about this in just a minute when we talk about facilitated diffusion, sometimes we get a little help with those proteins in the membrane and they form channels that actually will allow things that are charged or sometimes large to diffuse across the membrane down their concentration gradient. So that just gives you an example of some of the things that will diffuse down their concentration gradient in the human body. Now, osmosis, okay, is just a fancy word for water diffusion, okay? And so I think a lot of people get caught up in this term osmosis, and that's all it is water diffusion. This is talking specifically about water, okay, now diffusing down its concentration gradient. So when we talk about osmosis, okay, again, osmosis is concerned only with what's happening with water. So, for osmosis to occur, okay, we have to have a couple conditions. We have to have a, again, membrane that's semi-permeable, okay? So here's our, I'm drawing in red that semi-permeable membrane, okay? And so we'll pretend like, again, we've got this, big beaker, and I've just inserted this semi-permeable membrane. And what that means is that membrane can be permeable to water, okay, but is going to prevent, okay, other things like certain solutes from moving across that membrane. And we have to have that to create our concentration gradient of water, okay? So now let's talk about how this happens. In this case, okay, here we have our semi-permeable membrane, and in purple here, okay, we're going to have our solute, okay? So in this case, this is our solutes. And a solute, again, is just something that would dissolve in, again, water. So our solute would be something that would dissolve in water. So in this case here, okay, we can see, and we'll just say for, example's sake, let's say it's salt, okay? So for this case here, okay, if you look on this side of the membrane, okay, we have a lower solute concentration, okay, or a lower salt concentration. And on this side of the membrane, we have a higher, okay, solute concentration. And again, for an osmotic gradient to be created, okay, the solute, there has to be a solute that cannot cross the membrane. So in this case, our solutes, and again, we're using salt as an example, okay, cannot cross the membrane. So here our salt, again, molecules cannot cross the membrane. So now, okay, to talk about what happens with osmosis. So the easy way to look at osmosis is, again, we're concerned with where does water want to move down its concentration gradient, okay? And again, for osmosis to occur, this membrane has to allow water across but not solute. So in this case here, okay, we have lower, okay, solute, or in this case we'll just say salt. We have a lower salt concentration, right? Okay, and so we have fewer salt, again, we've basically fewer salts, okay, floating around the solution here. And so fewer salts, okay, floating around that solution leaves for more room for more water molecules, right? Okay, so literally, okay, we have more room for water because, again, we have fewer salts taking up space in here. On this side here, okay, we have a higher, okay, salt concentration in purple there. And so, again, because we have more salts, they're taking up more space. And that leaves us less room, right, for water. Okay? So now this has created our concentration gradient of water. Okay? So now water is in a higher concentration on this side and in a lower concentration on this side. Okay? And so now water is just going to very simply diffuse, as you're seeing here, down its concentration gradient from an area where it's in a higher concentration to an area where it's in a lower concentration until the water molecules on this side, okay, are equal to the water molecules on this side. So, okay, again, my easy trick for you in osmosis problems, okay, is to do this and break it down like this, okay? When you're looking at, for example, and mostly we're going to talk about cells being in certain solutions, okay? So I'm going to draw another beaker over here, okay? And inside my beaker, okay, we are going to have a cell here with, again, a plasma membrane, and that plasma membrane is semi-permeable, okay? And so when we look at this cell, okay, I'm going to draw my salt concentration out here, okay? And you're going to see, I'm going to make this a really high So we'll just use salt as an example again. Okay, really high salt or solute concentration. Okay, and then, okay, inside the cell, I'm going to make this a lower solute concentration. Okay, so, okay, again, the easiest way to do an osmosis problem like this, and what I would typically do is ask you, what's going to happen to my cell here? Okay, so the easiest way to walk yourself through this is to go, okay, my solution outside here, outside the cell, okay, here is going to have a higher solute concentration, okay? And if it has a higher solute concentration, that means then it has less room for water, okay? And now inside my cell here, okay, it's just the opposite, right? I have lower solute, okay, if you can read that I'm squishing it in there, okay? And that leaves me more room for water. And because that leaves me more room for water molecules, okay, then my water molecules are in a higher concentration inside the cell than they are outside the cell, okay? And so where's water going to move? Water is going to move right down its concentration gradient, okay, from the inside of the cell to the outside of the cell. And what's going to happen? Okay, my cell is going to shrink, okay? And that's exactly what would happen again if you put a cell and exposed it to a really saline solution. Okay, literally the cell would lose water to that surrounding salt solution. So this is the easiest way to go through these problems. Okay, if again you're going to mark what's on one side of the membrane versus the other. Okay, and whichever has the higher solute concentration is going to have less room for water, lower water concentration, and whatever has the lower solute concentration has more room for water. So again, water is just going to simply move right down its concentration gradients until the water molecules are equal on one side versus the other. Now, water molecules are actually always in motion. They're always moving, as are all molecules, according to what we call kind of random molecular movements. So molecules are always kind of bouncing around a little bit. And so that's what allows these molecules to move from one side to the other. So now there's this term called tonicity, okay? And tonicity just means if we have a certain solution, how will that solution affect, what type of effect will that solution have on cell volume? And that was kind of what we did, what we already did this example just a minute ago when we talked about the previous slide. I drew out the cell in the really saline solution. So an isotonic solution, okay, means that we have, iso actually means same, just so you know. So what this means is we have an equal solute concentration on both sides of the membrane. So again, you have the same concentration of these solutes that cannot cross the cell membrane, okay, on the inside of the cell as you do on the outside of the cell, okay. So isotonic would mean that you would have no what we call net movement of water molecules. If our solute concentrations are equal, okay, on both sides of the membrane, then the number of water molecules on both sides of the membrane, right, the concentrations of those water molecules are equal. So we'll have an equal number of water molecules moving, okay, across the membrane, because remember, water molecules again are always moving. It's just that we'll have no what we call net movement. The same number of water molecules moving across one side will be moving to the other side, okay? So we'll have no, what I call no net movement, okay? No net movement of water molecules. So the next example of a solution would be what we call a hypotonic solution. And a hypotonic, and these solutions are always going to refer to the solution surrounding the cell, okay? So what solution do you have surrounding the cell? in the case of a hypotonic solution, hypo is always going to mean low. So that prefix hypo means low, and that's going to be from here on out. So for those of you that have had medical terminology, this will help. But for those of you that haven't, hypo means low. So a hypotonic solution means that we're always going to have a lower solution of solute, okay, surrounding the cell. So a lower concentration of solutes, okay, outside the cell than inside the cell, okay. And this is going to create what we call a lower osmotic pressure. And so in this case here, okay, again, you would have, I'm just going to draw my beaker. Here's my cell, okay? And in this case here, in a hypotonic solution, it's just the opposite of what we talked about before. That would be where you have a higher solute concentration inside the cell and a lower solute concentration outside the cell. So that's your hypotonic solution. And so now, of course, hopefully, some of you can guess what would happen to the cell here, right, in this case. If we have lower solute, right, lower solute in the solution surrounding the cell, that leaves us more room for water. And so if water is in a higher concentration outside the cell than inside the cell, where's water going to move? You guessed it, down its concentration gradient into the cell. Okay? So that's what we call a hypotonic solution. Now a hypertonic solution was the example that I gave in the last slide when we talked about, again, putting a cell into a solution that has a higher concentration of solutes, okay, in comparison to the body cell. So again, a hypertonic solution is going to be just the opposite of this case, where we now have a cell that's been put into a solution with a higher solute concentration. Okay? And that's where, again, instead of the cell expanding like it would here, the cell would shrink. Okay? So again, in a hypotonic solution, our cell will swell as it takes in water, and in a hypotonic solution, okay, our cell will shrink as it loses water. Okay, and so again, this is just the slide again, same slide, kind of going through the same different comparisons. Again, isotonic solutions, hypotonic solutions, and hypertonic solutions. So this slide is actually giving you a practical example of this. So this is showing you if you take a red blood cell and you put it in an isotonic solution, okay? Again, that means that the number of solutes, or the concentration of solutes outside the cell, are equal to the concentration of solutes inside the cell. And so in this case here, okay, that means the water concentration inside and outside the cell is also equal. So an equal number of water molecules are going to enter and exit the cell, okay? There's no what we call net movement, and so there's no change in the cell volume, okay? And again, that's because an equal number of water molecules are going to be moving into and out of the cell. Now, again, when we put a cell in a hypotonic solution, so we take this red blood cell, here you can actually see this picture of what red blood cells would look like if you took them out and you put them in a hypotonic solution where the solute concentration outside the cell was lower, right? So if the solute concentration is low, I'm just going to put low S here, okay, for solute. So if the solute concentration is low, then that means that leaves us more room for water. And if the water concentration is higher outside the cell than inside the cell, it's going to move down its concentration gradient, and that cell is going to swell. Okay, so then, okay, in the last case here, this is a hypertonic solution. So we've taken a cell and we've put that red blood cell in a solution that has lots of solute, okay? And so in this case, okay, that means that if there's lots of solute, okay, so we're high solute here, then we have less, okay, room for water. So that means that water is in a lower concentration outside the cell than it is inside the cell. So sure enough, you guessed it, water is going to move down its concentration gradient from a higher concentration inside the cell to a lower concentration outside the cell. And sure enough, okay, that cell is going to shrink. So that's just a practical example of tonicity and osmosis. So again, make sure that you're familiar with osmosis. Osmosis is a really, really important concept, not just for the exam, but it's going to be really important that you understand osmosis for when we get to the kidneys. So make sure that if for some reason you're not quite getting osmosis and you're not quite sure what's going on with it, and you had a little bit of trouble with that section, please contact again me via email, via my Illuminate office hours, any method you can to get some help with osmosis. Now, we can also get a little assistance across the membrane, okay? And so there are these forms of what we call membrane transport that can be assisted. And the reason that we might need this is just kind of what I had mentioned earlier in the lecture, that sometimes we have molecules, okay, that can't make it across the membrane. And those would be molecules, okay, that are large and charged, okay? So molecules that are large and charged, okay, do not like to move across the membrane. And the reason being is that, again, we have that lipid bilayer. And so again, fatty acids of those fatty acid tails of the phospholipids are not going to want to allow things that are charged through, because the fatty acids are not charged. So since they're not charged, they're kind of snobbish and they don't like to interact with other molecules that are charged. Okay, so things that are charged, or if it's just really large and bulky, it's not going to make it through that lipid bilayer. So, okay, if you remember from the very beginning of the first lecture, okay, when you looked at the membrane, we had that, kind of sandwich lipid bilayer, right, with the phospholipids and the, again, fatty acid tails hanging down like that, okay, and then we had these big proteins that would go through the membrane. And these proteins, okay, are important because many of them are going to act as what we call carrier molecules. And they can actually help bring things that are large and charged into the membrane. So carrier-mediated transport, okay, is when we have one of these carrier molecules, and again, they're proteins. And so one of these carrier proteins will actually flip and change shapes to help drop, again, a larger charged molecule from one side of the membrane, okay, through to the other side. And this process can be what we call either active or passive, okay? Now, so far, just to be really clear, we've only talked about passive processes, okay? So diffusion is passive, osmosis is passive, okay? And what that means is passive transport means that there is no energy or no ATP required, okay? These are things that are just moving right down their concentration gradient, okay? Now, something that's active transport, okay, is going to require ATP. And what that means is these are going to be molecules that are going to be pumped actually against the concentration gradient. So they're going to be pumped from an area where they're in a small concentration to an area where they're in a high concentration. And so they're going to actually go against the gradient. So it's an uphill battle. And that's going to require ATP. Okay? So again, this is just another term, and I'm going to introduce active transport again later in the lecture, but since it's here, I want to make sure I introduce it. So again, when we talk about these carrier-mediated transport proteins, they can actually, some of them will just be passive and just be letting, again, things move down their concentration gradient. And some of them are going to actually require ATP, and they'll actually pump things against the concentration gradient. Okay? Okay, so there are three important characteristics to remember for each of these carrier-mediated transport proteins. Okay, the first one is that they have to have specificity. Okay, and what that means is that these membrane transport proteins are going to be very specific for letting just certain molecules across the membrane. So they're going to be specific for certain types of molecules. So for example, you may have, again, a membrane transporter that's very specific for just glucose, and it's only going to let glucose across the membrane. Okay? The second thing to remember is that because whatever that molecule is, okay, again, let's say in this case it's glucose, okay, that's moving across the membrane through these transport proteins, because they require that transport protein, okay, to get across the membrane, then the membrane can reach, or those proteins can reach what we call a saturation point. And what that means is they can only move glucose across at a certain rate, right? And then we only have so many of these glucose transporters. So, okay, no matter how much glucose we might have outside the cell, okay, here, we can only move that glucose across the membrane, okay, so fast until we hit a point where all of our protein transporters, okay, are full of glucose, okay, and working as fast as they can. So we hit what's called a saturation point, where literally, if you add more glucose on the outside of the membrane, right, okay, it should diffuse faster and faster, okay, across the membrane until you hit this saturation point, okay? And what the saturation point is that's the point at which all of our glucose transporters are full and moving glucose across the membrane, and they're working as fast as they can, okay? And so they're saturated, they're full of glucose right now. And so even if we add more glucose, okay, on this side, it's not going to matter. We're not going to get the glucose to transport across any faster because all of those protein transporters are full and passing a glucose across the membrane. And then the third one is that we can have, again, competition. So sometimes, okay, there are other molecules that will be moving across the membrane at the same transporter as well. So this particular protein transporter maybe doesn't just move glucose, but it moves another molecule as well. And sometimes your protein transporters will be specific for, two types of molecules. And so those molecules will compete for that little receptor slot on the membrane, okay? And so whichever molecule is in a greater concentration outside here, okay, is going to be the molecule that's more likely to bind to that receptor site. So that's what we call competition. Okay? So those are the three important characteristics to keep in mind for any membrane transport protein. So when, okay, and this is where I know there's a lot of terminology, so I'm going to try to kind of go slow here. When we talk about, okay, membrane transport, and we talked about that carrier-mediated transport, okay, there were two types of carrier-mediated transport, right? There was active and passive, okay? Now, the passive form of our carrier-mediated transport is by a process that we call facilitated diffusion, okay? So what this means is, again, we had to have a carrier-mediated transporter, right, assist us in getting across the membrane, but it's still diffusion, okay? So we're still diffusing down a concentration gradient. So, is ATP required? No. Okay? No ATP is required here. And so because of that, this is a passive process. Okay? So this is what we call passive transport. It's an example of passive transport. So again, this is where substances are still moving from a high concentration down their concentration gradient to where they're in a lower concentration. And that's a passive process. Okay, unlike the simple diffusion, like carbon dioxide and oxygen that just move right across the membrane, again, facilitated diffusion is where we have something that requires a carrier molecule. Okay, so this is something that's moving down its concentration gradient, but unfortunately, it can't get across the membrane because, you guessed it, it's large, or it's charged, or it's both, large and charged. Okay. So for example, glucose is a perfect example of this. Glucose is actually going to get into our cells by facilitated transport, okay? And so the reason being is that glucose is big and it's bulky. So it fits the large portion of this category here, okay? And so because glucose is so big and bulky, it's not going to just directly move across the membrane. And so it needs this carrier-mediated transport protein to get across. And so when it moves across the membrane, again, it's going to still be through diffusion. It's going to be diffusing down a concentration gradient. It just needs a little assistance via this protein transporter. So again, this is an example, and unfortunately I can't play the animations for you like I would be able to in a classroom setting, but you can actually go to these animations and play these animations on the web yourself. So you can, pause this, write down, you can go ahead and write down the web address and then hop on and play these animations for yourself. And some of these animations are really nice. And so this animation actually shows you this facilitated diffusion using a carrier-mediated transporter. And again, it shows you that you're getting diffusion down a concentration gradient. Okay. However, it's through this, again, carrier-mediated protein transporter. Now let's talk about active transport, right? Okay, so we kind of already have talked about this. So active transport, again, is another type of assisted transport, except for this time we're moving something against its concentration gradient, right? So we're moving something from an area where it's in a low, and I'm going to abbreviate, this is my abbreviation for concentration, okay? You'll see they use it in chemistry. From an area of low concentration, okay, to an area of high concentration, okay? So this is against a concentration gradient. Because this is against a concentration gradient, it is going to require not only a carrier molecule, but it's also going to require energy, or ATP, to drive this process. So again, active transport is the only type of transport, again, where we're going to require ATP. And it's the only time that we're moving something against a concentration gradient. And so secondary active transport is probably something I'm going to actually talk a little bit more about once we get further on in the semester, so I'm a little less concerned that you kind of get this now without giving you some examples of this. But this is just where we actually use a concentration gradient that we've set up actively. So we've actively pumped something across a membrane to set up this concentration gradient to establish this concentration gradient so that we can use it to bring other things across the membrane. Okay, but again, I'm going to have you just for now, for simplicity, we'll get into secondary active transport a little later on in the semester. I'm going to have you just focus on primary active transport for now. So, and again, this is just showing you this active transport, showing you that you're actually, again, pumping things across a concentration gradient from a low concentration to a high concentration. And again, it's going to require ATP. Okay, so we have our carrier-mediated transporter here, but in this case it needed ATP to pump again this molecule across its concentration gradient. And again, if you want to watch an animation, this is a great animation. You can go to this website and watch this animation showing you active transport. So again, it's just a nice, if you're a visual person, this might be a nice animation for you. So other types of what we call assisted transport are going to be more of what I call, and you're going to see, I call this bulk transport. Okay, and this is going to be literally moving things across the membrane in bulk. So this is what I call bulk transport. And in this case, okay, we're actually going to be moving things across the membrane in vesicles. So this is where we're literally either taking something into the cell or moving some material out of the cell that's wrapped in a membrane-bound vesicle. Okay? And so some examples of this are going to be endo, and sorry, I don't know why it likes to leave out letters every once in a while. So we have what's called endo, E-N-D, okay, O, so that's endocytosis, and endo meaning that we're literally bringing something into the cell. So endocytosis is where we're going to be moving substances into the cell, okay? So we can move fluids into the cell by, again, taking fluids in bulk in these vesicles, okay? And this is what we call cell drinking or pinocytosis. This is cell drinking. And then we have what's called phagocytosis, which is where we're taking in, again, oftentimes food particles and sometimes foreign substances. And this is more of typically what we call cell eating, phagocytosis. And then we have something called receptor-mediated endocytosis. And what that means is that, you know, if we have a cell here, okay, and we're going to take in a certain substance, oftentimes the cell will have little receptors on it, on the surface, and these little receptors will only bind, okay, to like a certain type of molecule, okay, and when it binds that certain type of molecule, then what it will do is it will actually pinch off all of those molecules. So eventually, once again, it's bound a bunch of those molecules, okay, and here would be my little receptors. And you can see a better picture of this in your book. Then eventually it will pinch off those receptors into this little vesicle. So basically eventually it'll grow these little, what we call pseudopods out, okay, like this, and eventually pinch off those molecules and allow them to move into the cell. Okay, and that's what we call receptor-mediated endocytosis. So again, we're probably going to use the terms phagocytosis and pinocytosis honestly more often than the receptor-mediated endocytosis. But I want you to know these are all examples of endocytosis, bringing things into the cell in vesicles. And I'm going to see actually, I don't know that I actually had unfortunately a good picture of this for you, but I do have again an animation for you here. But in endocytosis, again, endocytosis, you're going to be bringing something into the cell. And so basically, okay, what's going to happen is typically the cell, and let's say even in phagocytosis or pinocytosis, okay, the cell, oops, and that was a horrible drawing here, but the cell will actually basically pinch off things like food particles or bacteria sometimes into, so basically it'll surround that food particle and eventually pinch off that food particle, okay, into this little vesicle and release it into the cell. And that way then, okay, this food particle, or sometimes it could even be a bacteria, okay, a bacterial cell, is now released into the interior of the cell, but it's surrounded by this little piece of the cell membrane. Okay, so this is your membrane-bound vesicle now. And now, okay, that allows us to safely fuse something like a lysosome, okay, that has all these digestive enzymes. So that lysosome can then fuse with the membrane of that vesicle, and then now digest that either food particle, or it could digest that bacterium. So again, that's how endocytosis is going to happen, is literally by engulfing, okay, either fluid in penocytosis or engulfing food particles in phagocytosis. So, and then exocytosis is just the reverse process. Exocytosis is where we're going to literally be expelling things from the cell. Okay, and we kind of already talked a little bit about this when we talked about, again, secreting things from the cell, like when we talked about secreting insulin. So in this process, okay, the cell is actually going to do just the opposite. Okay, here our cell is actually going to get rid of something. So we have a vesicle here. Okay, let's say that vesicle is carrying insulin. Okay, and that vesicle is going to actually move towards the membrane. Okay, and eventually, okay, it will fuse with the membrane. And then what will happen is when it fuses with the membrane, okay, literally that vesicle then will add more membrane to the membrane, right? We're constantly kind of reshuffling the membrane around as we shorten the membrane by pinching things off in bits of the membrane, okay, to make vesicles to take things in. But then we also gain membrane back when we have vesicles, again, that are going to release things to the outside, and now that membrane of the vesicle is going to become part of the cell membrane. And so here you can see again, in this case, would have been insulin. Now, as that vesicle fuses with the membrane, then insulin can be released to the outer external portion of the cell, or what we would call the extracellular matrix in this case. So again, that's what we call exocytosis. That's how cells are actually able to, again, expel things. Sometimes they want to just secrete things. Sometimes they want to get rid of waste products. Okay, so that's how the cell is actually able to do that. And those were all examples of bulk transport. Okay? Now, one other example of a passive process that occurs that we haven't talked about is a process called filtration. And this is one that, again, we're really going to come back to later on in the semester when we do urinary system. And so it's going to be real important in urinary system that we're going to talk a lot about filtration. And filtration is when molecules are going to be forced through a filter using hydrostatic pressure, water pressure, okay? Where we create a situation where we have a really high water pressure on one side of the membrane versus the other. And we can actually force things across that filter or membrane, okay, using hydrostatic pressure. This is a process called filtration. And you're going to see we're going to use this again in the kidneys when we talk about the urinary system. And that's a passive process. It doesn't require ATP. So again, this is just kind of a summary of all of the different processes. We've got passive transport, okay? And so our examples of passive transport were simple diffusion, where we had oxygen and carbon dioxide just diffusing right across the membrane. We have facilitated diffusion, where we have, again, diffusion using a carrier-mediated transport protein. We have osmosis, which is diffusion of water across the membrane. And then we have filtration, where we're using hydrostatic pressure to push things across the membrane. Okay? So that's all passive transport. That's all, again, examples of transport where we're moving things down their concentration gradient from an area of high concentration down to an area of low concentration. Okay? And so, again, those are all active transport. Or, I mean, those are all passive transport. Active transport, remember, is where we're moving things against the concentration gradient from an area of low concentration, okay, again, to an area where it's in a higher concentration. So this is an uphill battle, and this requires ATP. Okay? So again, that was active transport. And then we had, again, these examples of bulk transport, okay? And bulk transport is where we're using vesicles. So we can use vesicles to bring things into the cell, endocytosis. So we can bring, again, fluids into the cell via penocytosis, and we can bring, again, food into the cell, or, again, other molecules into the cell in bulk using phagocytosis. And then we can get rid of things, okay, and by exocytosis, again. So we literally package things in vesicles and then release them at the cell membrane. All right, so that's it for membrane transport, okay? So we're going to kind of switch gears here from membrane transport. And again, please take time to go over membrane transport now, because like I said, later on in the semester, that's going to become really important. It's important that you understand phagocytosis for things like the immune system. And it's important, again, for you to understand osmosis when we, especially in filtration. Osmosis is going to be more important in your understanding of filtration when we get to the urinary system. So again, it's important that you understand those things. It's important that you understand active versus passive transport. And if you're having trouble with that, please, again, feel free to contact me by any of the methods that you have available to you. So this next part of the lecture, we're actually going to talk, and again, hopefully this is a little bit of a review for most of you, but we're going to talk about how cells divide, okay? And so first we're going to talk about what's called the cell cycle. And in the cell cycle here, and the reason that this is important, obviously, is that we're going to talk about cells have to undergo this process called mitosis. Okay, and for a cell to undergo mitosis, I should say a cell needs to undergo mitosis for two main reasons, okay, since we're doing our cell biology here. And the two main reasons would be for, one, growth, okay? So again, you know, for us to go from the single cell, a single cell right after sperm fused with egg, right, to the big multicellular organism that we are, the cells had to undergo lots and lots of mitosis, okay? And so that would be our example of growth. The other reason that cells are going to need to divide by mitosis is that they need to repair themselves, okay, because there was damage to a cell, or replace, okay, themselves. So again, for example, you're constantly, right, shedding these epithelial cells from your skin. And so obviously your skin's not just disappearing, right? So you actually, okay, eventually will, your cells are constantly undergoing mitosis, and you'll replace all of those cells that you've shed. So again, those are the two main reasons why a cell is going to undergo what we call mitosis. Now, when we talk about, okay, mitosis, you have to understand that typically for most cells, and again, some cells are going to spend more time in mitosis than others. Obviously, your epithelial cells are going to spend a lot more time in mitosis than your liver cells are. But again, cells are going to spend a very small portion of their cell cycle actually in mitosis, dividing. Most of their cell cycle is going to be in a phase that we call interphase. Okay? And during interphase, this is where a cell is going to carry out most of its kind of, you know, cell day-to-day functions. And it's also where it's going to prepare for mitosis. So during interphase, this is all interphase, okay, from here all the way, okay, over here. And interphase is going to include what we call G1 phase, S phase, and G2 phase. In the G1 phase, okay, this is what we call our first growth phase, hence G1. And this is where cells are actually going to start to duplicate some of their organelles. So they're going to make more organelles, they're going to make more cytoplasm, make more cytosol to get ready for dividing into two cells. Okay? In the S phase, this is called S phase or synthesis phase, and this is where your DNA is replicated or synthesized. So you're going to make a copy of your DNA here. Okay? And then in the G2 phase, this is your last cell growth phase. So this is your last ditch effort to make some extra enzymes and proteins, make, again, any extra cytoplasm that you need to right before the cell divides. Okay? And then when we get to mitosis, okay, mitosis is going to be the process by which your cell is going to divide. So again, this is where we're going to divvy up nuclear material, divvy up the 2 copies of DNA now that we have, okay, into 2 sides of the cell, and then that cell will pinch off into 2 cells, into 2 identical cells. Okay? And as you probably remember, mitosis has 4 major phases that we break it down into to make it easy to understand. And these 4 phases are prophase, metaphase, anaphase, and telophase. And so when we talk about mitosis, okay, mitosis is actually just talking about the division of our DNA material, our nuclear, what we call nuclear division, okay? Or how we're going to divvy up, okay, again, these 2 copies of the DNA. So that's what mitosis is all about. Mitosis is all about divvying up these 2 copies, identical copies of your DNA, into opposite sides of the cell. Okay? Cytokinesis, okay, is actually going to be the divvying up of all of the other stuff beside your DNA. So this is going to be how we divvy up the cytoplasm with all the organelles in it. So again, splitting up and dividing the cytoplasm and the organelles into the 2 different, again, sides of the cell, and eventually, okay, pinching that cell off into 2. That's cytokinesis. So I'll talk more about that as we kind of go through. So again, this is probably just reiterating some of the stuff I just talked about, but interphase, okay, is going to be that phase where we have the growth phases where we're making extra cytoplasm, we're making extra cytoplasmic material, so that way then we've got it ready for when we undergo our division. And again, it's also where we're going to replicate our DNA as well in preparation for cell division. So again, we have our G1 phase, which is our cell growth phase, our S phase, which is where we're making a copy of our DNA, right? Okay, and then our G2 phase is our kind of second and last ditch effort to, again, produce any enzymes, any proteins, any extra cytoplasm material that we need right before cell division. So then when we get into, again, mitosis, remember mitosis is just literally the division of the DNA inside the nucleus. So how we're going to divvy up the DNA inside the nucleus, okay? And so for our 4 phases, okay, to kind of help you remember our 4 phases, okay, our first phase is going to be prophase. Okay? Prophase, right, has a P in it, so to help you remember this is where we're going to pair up our sister chromatids, okay? And so the easiest way to identify prophase is this is going to be the first phase where you're actually going to get to see chromosomes. Remember, most of the time, inside your nucleus, your DNA is going to be in the form of chromatin. It's going to look like spaghetti, okay? And it's only when we undergo mitosis, especially prophase, that we're actually going to get to see these nice pretty chromosomes, okay? Now remember that we've made a duplicate copy of our DNA, and so to organize this DNA, we're going to form these nice pretty chromosomes that are actually going to be duplicate copies, believe it or not, okay? So here, when you actually look at this pretty chromosome, that's again a big X, what you're going to see is here you actually have, okay, here it would be one copy of your chromosome. Let's say this was chromosome 1 from your mom, okay? And you're going to see that here is going to be your duplicate copy, okay? Remember that in the S phase of interphase, you made a copy of all of your DNA, okay? So you are going to pair up those copies in this pretty chromosome, okay? And so literally, that's what a chromosome is. It's a duplicate copy of your DNA. And so the duplicate copies here, okay, we actually call these the sister chromatids. This one here, this side here, is identical to this side here, okay? And so I like to call these the identical sisters or the sister chromatids, okay? And you'll hear them more frequently called sister chromatids. Sister chromatids, okay? And they're just identical copies. And so remember that we actually have 23 pairs of chromosomes, right? One from mom and one from dad, okay? And so let's just say that this is chromosome 1, okay, out of 23, right? Here's chromosome 1 from mom, okay? That's the copy that you got from mom. And this over here, okay, was the copy that you got from dad, chromosome 1 from dad, okay? And so when you made a copy of that DNA, okay, and then in prophase, you're going to pair up these copies together. And when you form these pretty chromosomes, okay, you actually are going to have duplicate copies. So here you can see you've got chromosome 1 from mom, and then this is the copy, okay, attached to it. And same thing here, this would be chromosome 1 from dad. And again, you're going to have your copy attached to it. Okay, so again, that's what we call sister chromatids. Those are the identical copies. Now, those sister chromatids are going to be attached, okay, together at this area called the centromere. Okay, so where these duplicate copies are attached to each other, the sister chromatids are attached to each other. We call this the centromere. Okay, so again, in prophase, this will be the first phase where we're actually going to get to see our chromosomes, and so you'll get to see these paired sister chromatids, okay? Because prophase is the first phase where our DNA is going to actually condense, okay, from its loosey-goosey chromatin form into this nice, tight, folded, coiled DNA inside a chromosome. And again, our sister chromatids are going to pair up. Then in metaphase, okay, our chromosomes are actually going to line up in the middle of the cell, hence metaphase, midway, right? So we helped you to remember M and M. Our chromosomes are going to line up in the middle of the cell, okay? And then in anaphase, okay, we're actually going to pull apart these duplicate copies. We're going to pull apart these sister chromatids or pull them away from each other to the opposite sides of the cell, okay? And then, okay, in telophase, okay, this is where we're actually going to, our nuclear material, okay, will be pulled into each kind of half of the cell there, and you're going to see our DNA is going to do just the opposite of what it did in prophase. It's going to go from these nice tight chromosomes, and it's going to decondense into this uncoiled phase, and then at the end of that, then our cell will literally tear into two or split into two. Okay? So again, if you think of mitosis, mitosis is very easy. Okay? What you're doing in mitosis is literally you start with your chromosome, and just before mitosis, I should say in interphase, okay? And so in interphase here, okay, you have your DNA, okay, and again, you're going to make a copy of that DNA, a duplicate copy of that DNA, okay, and then, okay, you're going to go through mitosis, and you're going to split or pull apart, okay, these duplicate copies so that you end up having one copy on this side of the cell and one copy on the other side of the cell, and then eventually the cell will just pinch into two. That's how easy mitosis is. Okay? Now, quick question for you. Okay? DNA synthesis, true or false? DNA synthesis occurs during mitosis. Okay? And the answer to that is false. Remember that your DNA synthesis, or when you copy your DNA, actually happens before mitosis in interphase. Okay? So keep that in mind. And then cytokinesis, again, is actually where you're divvying up the cytoplasm. So cytokinesis actually happens at the same time that telophase happens here. And cytokinesis is where basically, again, if you remember, mitosis is all about divvying up your DNA. Cytokinesis is all about divvying up your cytosol and all of those organelles. So, you know, if you're taking one cell and dividing it into two, you got to divide up the mitochondria, okay? You have to divide up, again, the endoplasmic reticulum. So that's the process of cytokinesis. And that happens, again, alongside of the end of mitosis here. So again, this typically starts to occur during anaphase, and we'll finish up, again, in telophase. So again, towards the end of mitosis. So again, let's just go through each of those phases in detail here, and I've got some nice pictures here. So here you're going to see this is prophase, and during prophase, again, if you were to have a question on an exam about prophase, this will be the first time that you are seeing chromosomes, okay? And that's because the big thing that happens in prophase is that the chromatin is condensing and shortening into these pretty chromosomes, okay? And this is also where you're going to see your sister chromatids are paired. So again, in metaphase, okay? Now in metaphase, you're going to see here your chromosomes are lining up single file, okay, at what we call the metaphase plate, the midway region of the cell, okay? And what you're going to see here is, and again, let's just go with our example where we had, this is chromosome 1 from mom here, okay? And there's our duplicate copy, our sister chromatids, right? Okay, so there's chromosome 1 from mom, there's chromosome 1 from mom, duplicate copy. Here's chromosome 1 from dad, and here's our duplicate copy, chromosome 1 from dad, our sister chromatids. Okay, so here, and you can see this again, nice in this picture here, that during metaphase, our chromosomes are lining up in a way that it makes it very easy for our sister chromatids to be split in the next phase. So you're also going to see the formation of what we call the spindle apparatus. The spindle apparatus is where these structures here are what we call our centrioles, for those of you that might remember some of this. And okay, out of these centrioles, we're going to produce these microtubules. Remember, microtubules are important in cell division? And some of these microtubules are actually going to attach to these, again, chromosomes at the centromere. Okay? And that's actually how, in the next phase, they'll be able to pull them apart. So what ends up happening is that's actually how they get them to line up. Basically, these microtubules will lengthen and shorten, okay, until they get those chromosomes all lined up nice and pretty in the center of the cell. So here you can see in this picture here, you can see a really nice, and this is again an actual picture of a cell and cell division here. And this is a fish blastulus cell here. And if you look, not that you need to know that, it's just a fish egg. And we can look at mitosis really well because we get to see lots of cells that are undergoing mitosis in a fish egg. So what you can see here is here's the centrioles, okay? And then here's the microtubules growing out here to attach to those centromeres. And this structure here, this whole structure here, is what we call the spindle apparatus, okay? Because it forms this nice pretty spindle apparatus. We can see that nice, quite well in metaphase. So then in anaphase is actually our shortest phase of mitosis. And you can see in anaphase, this is where we're actually going to pull the sister chromatids apart from each other. So if we go back here again in this phase here, in anaphase, what's going to happen is these microtubules are going to shorten, and it's going to pull these sister chromatids in the opposite direction from each other. So in anaphase, we're actually pulling apart the sister chromatids. That's the big thing that's happening here. Okay, we're pulling apart the identical copies, or the sister chromatids, towards each opposite end of the pole. Okay. So again, you can see that, and it ends up looking just like this, where you see it looks like a bunch of these little V's, okay? And that's because the sister chromatids have been pulled to the opposite ends here at the centromeres as these microtubules shorten. And then in telophase, okay, this is where literally our cell is going to start to pinch off, and you're going to see in telophase what happens here is just the opposite of prophase. Okay, now the chromosomes on each side of the cell here, okay, are going to start to decondense and unravel into their nice thready-like form called chromatin. Okay. Remember now, we have identical copies here. We have identical copies on each side of the cell. And the big thing that's going to be your kind of indicator for telophase is you're going to see the formation of this thing right here called the cleavage furrow, where it pinches in, just like this. Okay. This is called the cleavage furrow. And if you remember, I talked about this a little bit in the first cell lecture when I talked about microfilaments. And microfilaments, actin, okay, filaments are going to wrap all the way around here, okay, to form this cleavage furrow. And what they're going to do is they're contractile filaments, so they're going to start to contract and pinch in, just as if you were releasing a rubber band, okay, around there. And it's going to cause those two cells to pinch in until eventually, okay, they literally pinch apart into two cells. So again, that's your basics for mitosis, the 4 different phases of mitosis. And again, if you have any questions, please feel free to ask me. Now, later in life, in life for a cell, eventually that cell is going to undergo what we call cell differentiation. And that's where cells basically are told what type of cell they're going to be when they grow up. So cells can actually become specialized for certain particular types of functions. And this makes sense when you think about, obviously, a liver cell is doing something very different than an epithelial cell, right? So a cell that would be, aligning your skin. So this is what we call cell differentiation, where cells become very specialized for certain functions. And so again, oftentimes we start out with, you guys have heard the term stem cells, right? And these stem cells are cells that still have the ability to undergo cell division. They're flexible and they can become, again, different cell types. And then eventually, okay, that cell will eventually be given a signal to differentiate and develop certain organelles, again, that are more specialized for certain functions. So again, if you're a liver cell, then you may start to produce more of that smooth endoplasmic reticulum because you're going to have this particular function of detoxification. So, and this oftentimes, again, is going to occur due to the expression of different genes. So you'll have certain genes inside that cell that will be turned on and expressed at a certain phase in a cell's life that are going to cause that cell to be specialized for certain functions. And then oftentimes that cell can also receive certain cell signals as well from the external environment that tell that cell what to be basically when it grows up. Apoptosis, okay, is going to be at the end of a cell's life, okay, and that's actually when a cell has basically programmed itself to die. So this is what we call programmed cell death. And we can see this in development, which is kind of interesting. So if you look at a fetus, at one point in the development of a fetus, you'll actually see webbing in between the fingers of a fetus. And eventually that webbing just disintegrates. And the reason that it disintegrates is that those cells are actually programmed, okay, during a certain phase of development to undergo apoptosis, or literally to undergo cell death. And so Again, that's an example of apoptosis. Also, if you've ever really, again, got a bad sunburn, okay, you've damaged that outer epithelial lining, then those cells will actually go ahead and be triggered to undergo apoptosis or programmed cell death. So literally when you, again, damage by getting a bad sunburn, that outer epithelial layer of your skin, okay, you're going to be triggering those cells to undergo apoptosis, to kill themselves. And that's why eventually that layer will slough off and you'll end up having to deal with that lovely process that we call peeling. All right, so the very last part of this lecture is probably, I think, going to be the tougher part of this lecture for some of you in that. And then some of you, again, it's going to be a review. So it just depends on whether or not you have had this material before. If you have not gone through this material before, then I suggest that you read this in your textbook first, because I'm only going to give you the highlights of what you really need to know on this process for kind of the rest of the semester. So again, I'm just going to be giving you kind of the highlights. But this is an important process, and it's important because we have to understand, if we're going to talk about cells and how cells function, we also have to understand how they process and produce energy. And the reason being is that that's how a cell functions. And if a cell can no longer process and produce energy, then that cell will eventually break down. Okay? So that's this last bit of the lecture is talking about how cells process and produce energy. So when we talk about energy production, we can break this really down into some major steps. We'll talk about glycolysis being the first step of energy production. The second step of energy production, and I'm just going to kind of go back and fill this in here. So again, just so that you know that we're breaking this down into 3 different steps. We're going to break this down into glycolysis, the citric acid cycle, okay, is going to be kind of the second step here. And the last step will be the electron transport chain. So that'll be kind of the 3 phases that we're going to break energy production down into. Now, when we talked about where energy production occurs inside the cell, okay, we talked about the fact that the mitochondria are actually the powerhouses for the cell. And so the citric acid cycle and the electron transport chain are actually going to happen. They're going to occur inside the mitochondria. Glycolysis is the only process, okay, that does not occur inside the mitochondria. It actually occurs out in the cytosol, okay, outside of the mitochondria. So glycolysis, we're not going to go through all of these reactions, don't worry, okay? But glycolysis is a complicated process of about 10 separate reactions that occur in sequence, okay? And again, this is the very beginning of energy production. So this is where we're actually going to, and again, we're not going to go through these 10 separate reactions. You can look in your book if it helps you to kind of go through that, but we're again primarily going to kind of give you the major material that you need to be able to be successful from here on out. So again, I'm going to be giving you just the highlights here, and that's what you're going to be responsible for in the exam. So in this first step of glycolysis, that again is just happening out there in the cytosol, okay, we're going to take one glucose molecule, okay, and that glucose molecule is going to be broken down into 2 molecules of pyruvic acid, or what you'll hear called pyruvate, okay. So basically, okay, what we're going to be doing is, and if you remember, glucose is a 6-carbon molecule, okay? Glucose is actually C6H12, okay, O6. And so what we're going to be doing is we're going to be taking this 6-carbon glucose molecule, okay, and we are going to break it down into two 3-carbon molecules. Okay, so we're basically going to be dividing that 6-carbon molecule in half, okay, into 2 molecules of this pyruvic acid. Each pyruvic acid has 3 carbons in it. And this process is what we call anaerobic. And what that means is that when we do this breakdown process, okay, it does not require oxygen. So no oxygen is needed here. And, okay, because we are breaking a chemical bond, we're actually breaking lots of chemical bonds here, but because we are breaking, okay, this chemical bond, there is energy that's trapped inside, okay, that chemical bond. And when we break that bond, okay, then energy will be released and will be captured in the form of ATP. So we're actually going to produce 2 ATP molecules of energy here. Okay? So at the end of glycolysis, okay, at the very beginning of glycolysis, we start with glucose. Okay? And at the end of glycolysis, we end up with 2 pyruvic acid, okay, and 2 ATP. So that's your kind of summary of glycolysis. And again, this actually happens in, there are actually 10 different steps in here by which this process happens. But again, you're only responsible for knowing this information. So then, what happens to these pyruvate, right? Again, obviously, we're going to be producing more energy than just 2 ATP. 2 ATP isn't a whole lot of energy, right? So, okay, this is where the citric acid cycle comes into play. Now, the citric acid cycle, okay, is going to actually occur inside the mitochondria, okay? So, this happens inside the mitochondria, not out in the cytosol like glycolysis, but in the mitochondria. And again, that's why the mitochondria, as you're going to see, is going to be called the powerhouse of the cell. You're going to see that the end product, okay, after we talk about the citric acid cycle and the electron transport chain, is going to be a heck of a lot more ATP than just 2 ATP. 2 ATP isn't a whole lot. So what's going to happen now is these pyruvates, okay, these pyruvic acids are going to be brought into, okay, the mitochondria. And when they're brought into the mitochondria, they're going to be converted into what we call acetyl-CoA, okay? And remember that each of these pyruvates actually had 3 carbons. If you look here, they actually draw the 3 carbons for you, okay? So again, from glycolysis, they tell you had 2 molecules of this pyruvate, right, per glucose that were produced. So before we go into the citric acid cycle here, the first step, okay, of the citric acid cycle is going to be to convert this pyruvate here into acetyl-CoA. Okay? And when it does that, we're going to be releasing or breaking one of these bonds and releasing one of those carbon molecules in the form of carbon dioxide. Okay, so you can see that here. And so we're going from a 3-carbon molecule to a 2-carbon molecule. Okay, we're going from pyruvate or pyruvic acid to acetyl-CoA. And so that carbon is released in the form of carbon dioxide. Now the cool part is, okay, this now tells you why we have to breathe out carbon dioxide as a waste product. It is a waste product of energy production inside your cells. Okay, so every time that your cells, again, are producing ATP through the citric acid cycle, they are releasing carbon dioxide. Think about it. All the cells in your body are constantly doing this, right? So they're constantly releasing lots of CO2. So now in this process of breaking down, okay, pyruvates into acetyl-CoA, right? We broke one of these carbon-carbon bonds, and again, we had energy trapped inside that bond, okay? And that energy was actually going to be passed on to this electron carrier, okay? And this electron carrier is going to be important because it is going to, we're going to come back to this, I should say, when we talk about the electron transport chain. Okay? So we're going to talk about these kind of electron carriers where energy is going to be passed on to. Okay? And it's going to be kind of storing energy for now that will eventually be used to make ATP. Okay? So again, that first step of the citric acid cycle is going to be where we have acetyl-CoA here, okay? And acetyl-CoA is actually going to combine with a molecule called oxaloacetate in the Krebs cycle, and that oxaloacetate is constantly recycled, okay? So here in the very beginning for the acetyl-CoA just to get into the Krebs cycle, it's going to combine with this oxaloacetate molecule, and I'm not really as concerned that you know oxaloacetate because that's getting a little probably more detailed than we need for you to know. But I do want you to know that that acetyl-CoA is what, again, is first dropped into the Krebs cycle. So then, and again, I told you that, sorry that, unfortunately, it's cutting this off again, that oxaloacetate, or oxaloacetic acid, the same thing as oxaloacetate, is going to be regenerated at the end of this cycle. So what that means is when the acetyl-CoA combines with that oxaloacetate, okay, at the very end of this cycle, it's going to end up with just oxaloacetate, which means that you have 2 carbons, okay, to get rid of. So, okay, as you're going to see in the Krebs cycle, you're going to break down, okay, the bonds of these 2 carbons, okay, that have attached to that oxaloacetate. And so you're going to have to release those 2 carbons in the form of carbon dioxide. Okay? And as you break, okay, those bonds and break those molecules down, of course, you are going to store more of this energy, okay, in the form of these electron carriers, okay, NADH and FADH2. Okay? And you're going to see you're going to actually produce quite a few of them. If you look here, you produce an NADH here, you produce 3 NADH here, and you produce another FADH2 here. Okay? And so, and that's all from just 1 pyruvate. Remember, for breaking down each glucose, you produce 2, okay, pyruvate. So, hey, you're actually going to get a lot of these electron carriers, NADH and FADH2, okay? So again, and this is what this statement is talking about here. Hydrogen atoms, okay? And that's what you're seeing here. Hydrogen atoms, and we call them electron carriers because these hydrogen atoms contain electrons. And so hydrogen atoms are going to be released at certain points in the pathway. And these hydrogen atoms are going to bind to these hydrogen carrier molecules, okay? NAD and FAD to form NADH, okay, when they bind that hydrogen, NADH, and FADH2. So, and without getting into too much of the chemistry of this, guys, and I know this is hard, this part I think is the most difficult for students to understand because we don't have time to get into the chemistry of it, but I just want you to picture the basic idea here is that we're breaking down these chemical bonds of pyruvate, right, that were originally pyruvate and acetyl-CoA. We're breaking down these chemical bonds, and the energy that was stored in those bonds, okay, is now going to be released to these carrier molecules in the form of electrons. And again, these electrons are carried in these hydrogen atoms, okay? So that's basically how our energy is passed on. And now these carrier molecules are going to be carrying, okay, this hydrogen, okay, that electron in the form of hydrogen. And then we're going to talk about what we use that hydrogen for here in just a minute. So if you look here, okay, in this process, okay, directly, okay, we'll produce 1 ATP directly per acetyl-CoA. Or remember that again, for each glucose, right? So we're getting 2 pyruvate and we'd get a 2 acetyl-CoA. So technically, per glucose, we would get 2 direct ATP, okay? And that's still not a whole lot, right, of ATP. We've only produced so far 2 ATP in glycolysis and 2 ATP directly in the Krebs cycle. But, okay, now you have to keep in mind that most of our energy, okay, is wrapped up, okay, in these carrier molecules, these hydrogen or electron carrier molecules. So now let's talk about how we get the energy out of those, okay? And that's where the electron transport chain comes in. And so again, remember, just like the Krebs cycle was inside the mitochondria, okay, the electron transport chain is also inside the mitochondria. And it's actually on the inner mitochondrial membrane, okay? So literally, this would be the inner mitochondrial membrane, and this would be the matrix, okay, in here. So again, remember the matrix was, if I could draw this up here, here would be your mitochondria. Here's that inner mitochondrial membrane, okay? And then here's the matrix inside here. So now what's going to happen is these, again, electron carriers, okay? So NADH, right? Okay? And FADH, NADH, and FADH2 are going to now release, okay, their electrons by releasing the hydrogen atoms, okay, so, or hydrogen ions, I'm sorry. So basically they're going to release their electrons, okay, by releasing these hydrogen ions. And so again, this is where they say the electrons are going to be extracted from the hydrogens, okay, that were transferred by these lovely hydrogen carriers, okay? And the energy that's going to be released by this transfer, okay, of electrons, so as we dump those electrons from these basically these hydrogen carriers, as we dump these electrons, these hydrogen ions, okay, are then going to be transported across the membrane using the energy, okay, from the electrons that were taken from those hydrogens. And so again, what's going to happen now is the energy released by that transfer of electrons, okay, is going to transport these hydrogen ions, okay, now from the matrix, okay, to this intermediate space, this space again in between these membranes here. And as it does that, okay, we're basically going to build up a hydrogen ion gradient here, okay? So we're going to create what we call a chemiosmotic gradient, okay? And so they're not actually, in this picture, they're not actually showing you this process. But what has happened is basically we have used, okay, these hydrogen ions, okay, that have, again, been released here, okay, to build up, okay, basically build up this, what we call chemiosmotic gradient, okay? And now, okay, we're going to use this gradient of hydrogen ions here that we've created, okay, to actually drive the synthesis of ATP. So now, okay, these hydrogen ions are going to flow down their concentration gradient through, okay, one of these protein transporters, and this specific protein transporter is called ATP synthase. And as you can guess, it's because it synthesizes ATP. And so what's going to happen is these hydrogen ions will move down their concentration gradient through this molecule. And as they move down their concentration gradient through this molecule is actually going to spin, okay? And as it does that, okay, it's actually going to drive the synthesis of ATP. So here you can see again we have an ADP, adenosine diphosphate, and then another phosphate group here. And as these hydrogen ions move down their concentration gradient, we are going to make ATP at an ADP and that phosphate group. Okay? So this gradient, this hydrogen ion gradient that was created by these carrier molecules, okay, is what's used to drive our ATP synthesis, okay, making ATP. Now, in the process we have, and it doesn't show you that in this picture, but we end up with these electrons at the very end of the electron transport chain, and we need something to catch those electrons. And so we actually have oxygen, okay, at the very end of the electron transport chain to actually accept the electrons that were originally dumped by these two carrier molecules. So again, this is a little bit of an involved process, and we're really oversimplifying it, which is going to be what makes this so hard to understand. But without, again, taking too much time and getting too involved in this, really what I want you to know is just the basics here. That here we have these, again, hydrogen carrier, okay, molecules. Electrons are going to be extracted from these hydrogens and these hydrogen carrier molecules, and then transferred through this series of carrier molecules, which you don't see here. You don't get to see that in this picture. And eventually energy will be released, okay, from this transferring of electrons, okay, and your book actually has a nice kind of picture of this. And as that energy is released through that transferring of electrons, it's going to transport hydrogen ions, okay, so that they, again, are going to build up in that intermediate space. And then the concentration gradient of those hydrogen ions is then going to be used to make ATP, okay? So that's basically the gist of what we're going to talk about for the electron transport chain. Now, the cell, okay, requires oxygen for the electron transport chain, okay? And so for this process to occur, we need oxygen, okay? So that's what makes that the aerobic process, okay? So the cell makes more ATP aerobically, okay? And this is going to be, again, when we talk about the citric acid cycle, okay, and the electron transport chain, because they're directly connected, the citric acid cycle and the electron, I'm going to abbreviate this, ETC, electron transport chain, are going to require oxygen, okay? And so the cell is going to make a heck of a lot more ATP aerobically than it's going to anaerobically. And the reason being is that we're going to get most of our ATP out of, okay, the electron transport chain. So you're going to see that with oxygen, okay, as our final electron acceptor, okay, we can actually make 34 ATP, okay, aerobically per glucose molecule. And I'm going to show you how this works here in just a minute. And then remember, there were only a net of 2 ATP that were made per glucose via glycolysis. And that's the part that does not require oxygen. So, okay, and remember this ATP, okay, that's produced is going to be used to make things in the cell. It's going to be used to transport things in the cell and to do mechanical work for the cell. So let me kind of summarize. I'm going to give you a nice little summary pathway of what would happen in terms of energy production. So here you can see in glycolysis, again, this happens out in the cytosol, right? So the cell, this is where glycolysis happens, out in the cytosol. We're going to make 2 ATP directly and 2 pyruvate, or pyruvic acid, same thing. Those 2 pyruvates are then going to be sent to the mitochondria, okay, where the citric acid cycle happens. And in the citric acid cycle of the mitochondria, okay, this is going to happen in the matrix here. And what's going to happen is we're going to produce, again, 2 ATP, not a whole lot of ATP, but look at this. We're going to produce 9 NADH and 2 FADH2. Lots of these electron carriers, okay? And then these electron carriers are then going to, again, dump their electrons, okay? They're basically going to dump their electrons in the forms of these hydrogen ions, okay, at the electron transport chain. And when they dump those electrons, okay, again, the energy from those electrons is going to be used to pump hydrogen out across from inside the matrix to the outside, okay, of that inner membrane, into that inner membrane space, where we're going to get this big concentration gradient of hydrogen ions. And then that lovely concentration gradient of hydrogen ions is then going to be used to make ATP, okay? And we're actually going to make a total of 32, okay, ATP from those 9 NADH and those 2 FADH2, okay? So we're actually going to make a lot of ATP. So again, that just gives you an idea of how we end up making so much ATP. So again, this 32 ATP has all come from setting up that concentration gradient of hydrogen ions from these 2 carrier molecules, NADH and FADH2. Okay, so NADH, FADH2 dump their electrons in the electron transport chain, and boom, we can make 32 ATP. So if we talk about the whole process, right, all together, we total it all up, okay, our total here is going to be 32 ATP from the electron transport chain plus 32 ATP from the citric acid cycle, right? That gives us 34, plus 2 more from glycolysis. So that gives us a total of about 36 ATP, okay? So again, you'll get to see that here, okay, as well. So again, you get that total of anywhere from, basically you'll get a total of about 36 ATP, okay? And you're going to see that some books are going to vary and say, you know, 36 to 38 ATP, and that's a little more involved. If you've taken BIO181, we talk a little bit about why that is. It has to do with this special carrier molecule here. But please don't worry about that for this class. That's beyond the level of what we're going to be doing for this class. So again, you're just responsible for knowing that, again, on average, we get 36 ATP out of breaking down a glucose molecule. So again, those are the processes that are involved. I think by far the most challenging is, again, kind of understanding energy production and the electron transport chain. Please read through that section in your book if you have not done the electron transport chain before. And again, keep in mind that we're going to be very basic for that. And I'm not going to go into any more detail on an exam than what we have in that slide. So again, it would be very basic. I really just want you to know for that electron transport chain that the electrons from those electron carrying molecules are used to create this hydrogen ion gradient, and that those hydrogen ions, that hydrogen ion gradient is then used to synthesize ATP. Okay, so hopefully that will help. Again, ease your fears. That usually tends to be the toughest section of this lecture. And like I said, please feel free to contact me via email. You can contact me via the Illuminate office hours, however you need to be able to, again, get through some of this material. So that concludes our cell lecture, and I will see you back for the skin lecture, which will be the last lecture before the exam.